Thank you. Good morning.
There you are.
This is Nick Dhurandhar. I appreciate the opportunity to present today, and I wish I were there in person, but the weather had different plans. So by way of conflict of interest, I should mention that I'm not presenting that work here, but somewhat related work has been, intellectual property has been protected, and there's a company that has funded part of that work, but it's not related. This is not a main focus of this presentation. So let's get started. Infectobesity is the word I like to use to describe obesity of infectious origin. Next slide, please. Yeah. And it is very hard for me to describe the entire field of infectobesity in the time available today, but what I'm trying to do is underscore some significant key salient highlights of this area under the attributes of the concept, animal models, mechanism of action, human studies, and then finally significance and implications. Next slide, please. So let's get started with the concept itself. And the question would be, is obesity an infectious disease? And this question, next slide, this question sounds similar to a question that was asked many years ago: is gastric ulcer an infectious disease? In fact, when I was in medical school in India, and the emphasis is on India, we were taught in medical school the causes of gastric ulcer, next slide, are hurry, worry, and curry. And that's to describe, of course, the anxiety or spicy food and so on. But of course, next slide. But of course, we know better now that the role of a bacteria, Helicobacter pylori, as a cause of gastric ulcer, thanks to the efforts of these, mainly to these gentlemen. So that was gastric ulcer, and now we are talking about obesity. Next slide. And there are a bunch of adipogenic pathogens that have been reported to cause, here is a list, they have been reported to cause obesity in various animal models, starting with the first one was in 1982, reported in Science. That was canine distemper virus that causes obesity in mice. And then there are several other pathogens, including viruses, scrapie agents, bacteria, or even parasites, causing obesity in different animal models that they have used. And you will notice a bunch of adenoviruses there, number 5, 6, 7, 8. We and my colleague and I were the first one to describe adipogenic properties of the first adenovirus, SMAM1, SMAM1. That's an avian adenovirus. And we also described adipogenic properties of a human adenovirus called AD36 or AD36 for short form. That was followed by other research groups discovering additional adipogenic adenoviruses, such as adeno37 and 5. And then the last two are associations, chlamydia and oral bacteria are associations, not causations, with obesity. So for the purpose of this presentation, I'm going to focus on AD36 to explain various attributes of infectobesity, and mainly because that is probably the most studied microbe for its adipogenic properties. So next slide, please. Human adenovirus 36 is one of the 50+ human adenoviruses that we know of, but it is antigenically unique compared to the rest of the group. It was first time isolated in Germany in late 70s from fecal sample of a girl suffering from diarrhea. So that is our only clue, perhaps, that this virus might be responsible for causing GI disturbances. And in our animal, in our hands, our animals, we didn't see any overt symptoms when they were experimentally infected. We know its entire genome, 32 kb, and its genes are divided primarily into two groups, early and late genes, denoted by the letters E or the L. Next slide, please. Now, talking about animal models, these are some experiments that we have done using various animal models, such as, let's go to the next slide. Yeah, the slide with red and blue bars. You will see there are animal experiments done with chickens, mice, rats, and marmosets. Those are non-human primates, weigh about 300 grams. And in every case, the uninfected control, that's the blue bar, compared to that, you will see significantly greater prevalence of obesity in the group that was infected with adenovirus 36. And that prevalence ranged in the infected group from 60 to 100%, depending on the experiment. Next slide, please. And here is an experiment as an example. These are marmosets. Uninfected control are blue color, and infected ones are red color, which gained about threefold greater weight in 6 months after their infection compared to the controls. And their body had significantly greater body fat as well at the end of those 6 months. Next slide, please. Here is another experiment done with mice that were on chow-fed diet. Now, there were 3 groups of mice. One was the uninfected control. The other group was infected with adenovirus 2, which is a non-adipogenic adenovirus, but just used as a control for infection. And the third group was infected with adeno 36. And the group infected with adeno 36 gained more body weight, more body fat. But the point I wanted to make on this slide is look at the glucose levels of 8036 infected mice. They started dropping, and so did insulin levels. So both graphs taken together tell us that there was a significant improvement in glycemic control in the animals that were infected with 8036. Next slide, please. And this is a very similar experiment, 3 groups of animals, again, same 3 groups, but these were animals fed high-fat diet. And it turns out that those animals that are infected with 8036 seem to be somewhat protected in terms of fat accumulation in the liver. And if you look at that panel of images, on your extreme left is a liver from a chow-fed mouse, which is nice and clean. The pink color is glycogen. White color is where lipid would have been accumulated, a very little lipid. And the rest of the other three images on your right side are from the high-fat fed group. So there will be, by definition, more fat accumulated, more white color in these livers. But between those, the one that was infected with 8036 had significantly lower lipid accumulation compared to mock-infected or adenovirus 2 infected mice. Next slide, please. Dr. Jae-won Nam from South Korea and his group have done, these are not our data, these are their data, have done a series of studies using 8036. And one of the things they discovered is that 8036 requires the inflammatory cytokine MCP1. If you use knockout MCP1 knockout mice, they don't respond to 8036 in terms of gaining adiposity. Next slide, please. And the next experiment they performed, they used an anti-inflammatory agent, mulberry extract, and observed that mulberry extract can attenuate 8036 induced obesity. And they have done many experiments, but I've just selected just a few to make a few points. In the next slide, they also use a ultraviolet light inactivated adenovirus 36 to prepare a so-called vaccine. And when they challenged vaccinated animals with 8036, they demonstrated they could prevent the onset of obesity in animals by using that as a vaccine. Next slide, please. Yeah. Some more points to mention about 8036 induced obesity would be that, as I mentioned, there are no overt symptoms that we have observed, no unintentional mortality observed in our hands in these animals. We have observed horizontal transmission to cage mates. So if you infect an animal and keep another uninfected animal in the same cage, you can that uninfected animal can acquire infection as well as obesity. This horizontal transmission. Another experiment that we conducted, we infected animals, obtained their blood in 36 hours, and injected that blood into the veins of a new set of animals. So we were mimicking blood transfusion from infected to uninfected animals. And we demonstrated that the uninfected animals who received that infected blood gets the virus and gets obesity as well. So fulfilling one of the Koch's postulate here, and these are all published. I just don't have the time to show all these experiments. And overall, there is not much impact on food intake. This obesity cannot be attributed, at least I don't think so, to food intake. And in terms of activity, we have not measured activity very objectively, but we didn't observe any effect on activity in those animals which are in the cage. Next slide, please. So moving on to mechanism of action. Next slide, please. And there are a bunch of studies, a lot of studies, many of them we have conducted, and then there are others who have conducted. And if I were to summarize all of those, let me just make a few statements. Next slide, please. So it looks like the mechanism of action for adipose tissue expansion seems to be by increasing proliferation, commitment, differentiation, and lipid accumulation in adipocyte progenitors by this virus, or ASC stands for adipose tissue-derived stem cells or stromal vascular cells. And next slide, please. Here is an example. These are 3T3L1 adipocytes, pre-adipocyte that differentiate into adipocytes. On your right side is the uninfected control, left side is the uninfected control, right side is the infected group. Green color is Bo-DP stain, which is lipid specific, and you will see much more lipid on your right side that were cells that were infected with 8036. Next slide, please. This effect of 8036 on lipid accumulation is dose dependent. If you look at the top graph, more virus to a point means more lipid accumulation. And the bottom half shows you that an antiviral drug, cidofovir, can actually kill or block this virus, attenuate the infection, and also reduce or attenuate lipid accumulation in these cells. Next slide, please. This lipid accumulation in these different pre-adipocytes appears to be very infection specific. Here is an image. All four images are of the same field. The blue color is DAPI stain, so it shows you nuclei of cells in this field, how many cells were there. The red color is a cell that was infected with a virus. That's a viral protein. Green color is Bo-DP stain, so it's lipid accumulation. And when you merge all those images, that is the bottom right-hand side image, you will see that lipid is accumulated only in the cell that was infected with this virus. So it seems to be a very specific effect on lipid accumulation in these cells. And these are human adipose tissue-derived stem cells obtained by adipose tissue biopsy. Next slide, please. And if we were to put in lots of experiments together, here is a working model in terms of cell signaling, what this virus appears to be doing. On the one hand, it appears to increase cyclic AMP pathway by CRE, that is cyclic AMP response element binding protein pathway, and on the other hand, PKB pathway or AKT pathway is activated. Together, the cells seem to, 8036 seems to increase proliferation of these adipocyte progenitors, leading to their differentiation and lipid accumulation as determined by various markers of adipogenesis such as CEBP beta, p-power gamma, as well as lipid accumulation that's on your right-hand side. Next slide, please. Another interesting observation to note is biopsied material adipose tissue from humans. When we took small pieces of this adipose tissue biopsy and cultured them as explants and infected them with the virus 8036, those infected groups, whether the biopsies were obtained from diabetic individuals or non-diabetic individuals, showed significantly greater glucose uptake. That's the upper panel in 8036 infected group. The lower panel shows similar finding, a dose-dependent glucose uptake increase in human skeletal muscle cells obtained by skeletal muscle biopsy from humans. A very similar effect. So 8036 appears to increase glucose uptake by this adipose tissue as well as human skeletal muscle cells. So next slide, please. So putting together data that I have shown and not shown, our working model looks like something like this. That 8036 on top left-hand corner of your screen, 8036 appears to infect adipocyte progenitors and increase their commitment and differentiation and increase glucose uptake inside the cell. This glucose is and then there is increased de novo lipogenesis, leading to greater lipid accumulation. So you have more adipocytes that have more lipids, and they are taking in more glucose. That may explain increased adiposity as well as enhanced glucose clearance from systemic circulation. Next slide, please. We have observed furthermore that of those various genes, the early gene 4, and its open reading frame 1, so E4 ORF, ORF1, that's the gene that is necessary and sufficient for adipogenic effect of 8036, certainly in vitro. Next slide, please. So moving on to human studies here. Next slide. Now, a million-dollar question is, do certain infections cause human obesity? Because up until now, I was showing all the animal models where we certainly see a cause and effect relationship between experimental viral infection and obesity in these animals. But what about humans? Now, of course, there are challenges. And you will notice all these challenges, to me, they sound like challenges that are facing this environmental pollutants area when it comes to determining their effect in humans. And one of the challenges is that obesity has insidious onset that we know. So if somebody is obese today, that person may not remember what happened 3 years ago, 5 years ago, when the person started becoming obese, what infection the person got. So that's number one issue. It's a different thing where a child gets sick, a perfectly normal child gets sick, goes to the hospital, stays in the hospital for a few days, and comes out and is diabetic. Now you can, or type 1 diabetes. So then you can start wondering about the connection there. But it's very hard in case of obesity. Number two is there are multiple etiological factors for obesity. So it is very difficult to attribute to one factor. You may identify people who have infection, a certain antigenic infection in the past, but your control group can still be obese because of some other reason. They don't have to have that particular factor. And then number 3 could be a combination of infection with other factors may be needed. So it may not be enough, depending on which infection you're talking about. There may be 2 or 3 things or stars need to line up for that obesity to show up. And finally, most importantly, ethical reasons preclude experimental infection of humans. So we're not going to be able to ever show direct cause and effect relationships between an infection and obesity, probably. But to my mind, it is similar to the association between smoking and lung cancer. To my knowledge, there is no study done directly to see if but some people start smoking and see what happens if they come down with lung cancer or not, and then there is control group. However, we have weight of indirect evidence is so strong that there is no doubt now whether there is a link between smoking and cancer. So we set out to obtain similar indirect evidence so that we can determine this question, do infections cause obesity? Next slide, please, human obesity. And we looked at natural exposure of humans to 8036. So if they were, we look for neutralizing antibodies to 8036. If antibodies are present, that could mean that sometime in the past they were infected. And this is our very first experiment with only 505 subjects. And it turns out that there were about 30% of these 500 obese, but only 11% of the non-obese individuals were positive for 8036. They were infected with 8036 sometime in the past. Moreover, those who were 8036 positive had significantly greater BMI compared to their seronegative counterparts. That was true for obese group as well as non-obese group. So even within the non-obese group, if you were antibody positive, you were likely to have, to be heavier compared to antibody negative non-obese individuals. We also screened them for other adenoviruses. They are non-adipogenic adenoviruses, adeno2 and adeno31, and those virus antibodies did not show this relationship at all. A different study, another cross-sectional study using twins. Now, we took advantage of the fact that body weight of twins is similar, if not the same, of adult twins. So our hypothesis was, if we find twins, that one twin is positive for the virus and one is not, we would think that the seropositive twin, co-twin, will be heavier compared to its seronegative counterpart. So we started out with 90 plus twin pairs, their serum sample, 180 some subjects, and focused only on discordant twin pairs. So if they were concordant twin pairs, both were seropositive or both were negative, we removed them and only focused on one positive, one negative, and there were 20 monozygotic, 6 dizygotic such twin pairs that we found. Next slide, please. And it turns out that in these 26 discordant twin pairs, indeed, those that were antibody positive were significantly heavier and significantly fatter compared to their antibody negative counterparts. I think this is the closest one can come to determining the role of 8036 in human obesity, short of infecting them. Of course, we do need more sample size, and that would be super to further test this. Next slide, please. Another study, cross-sectional association in 1,500 Caucasians, Hispanic, African-American men, women, and children screened for the presence of 8036. Turns out that this 8036 infection was indeed associated with better glycemic control and lower hepatic lipid levels. If you remember, mice responded by having some sort of a protective effect on their liver when they were infected with 8036, with a similar picture, but this is an association. These human studies, of course, an association, not a causation. And Trovato, Guglielmo Trovato in Italy and his group has done a series of experiments with 8036, and within humans studies, they have shown very similar findings in their hands, including actually less prevalence or sort of protective effect on non-alcoholic fatty liver disease in people who are infected with 8036. So that kind of fits in with these findings as well. Next slide, please. Now, this is a longitudinal study, a 10-year prospective study of 1,400 men and women screened for exposure to 8036. So those who were seropositive at baseline for 8036 antibodies had significantly greater increase in body fat 10 years later, but had significantly less decline in glucose control, glycemic control, compared to their antibody negative counterparts. Next slide, please. These are not our data, but they are from Sweden, and these are different populations studied at different time points. It's not the same population, but these are so studied in 1992, 2002, 2004, 4 to 6, or 2009, and these are lean subjects, and 8036 antibody prevalence appears to be greater in these populations as time passes by. Again, this is not the same population study. There are different groups studied over time, but in the same country, in Sweden. During the same time, prevalence of obesity increased in Sweden in men and women. Next slide, please.
Dr. Dhurandhar, we're going to need for you to wrap up at this point, if you could. Thank you.
Okay. Next slide, please. I thought we started at 8:50, right?
We started a little early, actually. That's why. But go ahead and.
Okay. All right. So these are what you're looking at is a review. At that time, there were 12 studies in humans. Of those, 10 showed a significant association of 8036 infection with obesity, and 2 did not. Next slide, please. And at today, almost every continent the researchers have reported presence of 8036 and association, except for Africa. There has been no report that I know of. Next slide, please. There have been meta-analyses done. There is one here that shows increased odds ratio of being obese if you have 8036 antibodies. And next slide. And there is another meta-analysis done, very similar findings. And finally, one more study we conducted in samples of Pima Indian adipose tissue samples received from NIH facility in Phoenix. It showed that those subjects that were positive for 8036 DNA, about 27% of them were, had about 8-fold greater ability for the viruses, for their pre-adipocyte to differentiate into adipocytes. Slide, please. Not all, and there are some studies such as this study, such as one from University of Georgia that showed that 8036 antibodies presence correlated positively with adiposity, but negatively with bone strength. Next slide, please. And in this study with children, it showed that 8036 positive children lose less weight when they're attempting to lose weight. Next slide, please. So finally, significance and implications. Next slide. Next slide. But I submit that obesity is a complex disease with multifactorial etiology. And if this is a disease with multifactorial etiology, we need a multifactorial treatment and prevention approach. And that may give us better results. Next slide, please. So specifically speaking about infectobesity, it may have unique prevention and treatment strategies for instance, something like 8036 induced obesity. And a vaccine may be perhaps easier than behavioral change that we are trying to induce. And we all know how difficult that is. And think of smallpox that was eradicated with a vaccine. And eliminating 8036 from environment may not be needed if we have a vaccine to protect individuals. I do want to mention here two things, that by no means I'm suggesting that all obesity is due to infection or all obesity is due to adenoviruses. Next slide, please. We know that infection with 8036 causes obesity in animals and correlated with human obesity. And there may be, if there are few adipogenic pathogens that have been identified, imagine how many may be out there awaiting discovery. So the question is, have infections contributed to the increase of prevalence of obesity since 1980? I do not know answer to that question, but that is a question to ask. Next slide, please. Now I want to, for the end of just very quickly, I want to go over some, it's very difficult to do remotely. I would have done it quicker myself, but I'll try. So I want to go over real quickly CDC slides about prevalence, and I want to provide some other question, not the same thing that you have seen before. Asthma, influenza, and obesity. So let's get started. Next slide, please. So these are slides of prevalence, about CDC slides about prevalence of asthma. And we'll go quickly, as after I mentioned, we'll go quickly through these slides, and you will notice that there is this over time, there is a random pattern, no particular pattern for increasing prevalence or spreading of prevalence, because this is a non-infectious condition. So if you go through 2-3 slides real quick, next slide, like just click real quick, you go through those. Asthma prevalence till 2007. Yeah. Okay, you can stop here now at those slides with influenza. The next slide is flu. So if we come to the slide with flu or influenza prevalence, now this is a slide that has spread of influenza in the United States. And as you would imagine, an infectious disease is spreading as it would to neighboring states, and then it develops a focal point and it goes around spreading in those states. So let's just go through next. Aaron, please go through those slides quickly as you just keep clicking, and you will see that influenza is spreading in neighboring states, and the yellow color appears. That is a deepening of disease prevalence, and darker, and it's spreading to its neighboring state as a disease would. And if you go to the slide that says obesity, and now you will notice, we're going to look at those slides that you all have looked at 1,000 times before, but keep this in mind and look at the spread of obesity in various states. And Erin, if you please start clicking those on those slides, And you will start seeing that the color, as particularly when the deeper color appears, you will notice that. Now look at the blue color, dark blue color, and how this prevalence is spreading in neighboring states. And now the yellow color, or the cream color, there you go. And you see the red color appearing in neighboring states and filling up. Next would be North Dakota and Minnesota, and so on. So this is a pattern of obesity spreading through the United States. And next slide, please, Aaron. So I want to leave you all with this question that says, why does the spread of obesity in the United States resemble an infectious disease? I do not know. I truly do not know answer to that question, but to me it does look like an infectious disease spreading through the United States. So next slide, please. And with that thought, I would like to say thank you very much for your attention. And what I'm pointing out is how much work remains to be done in this area. Thanks again.
Thank you so much for that. We, just to stay on our schedule, we're going to have about 4 minutes for any questions or clarification. And I actually have a question for you. And I'm sorry, by the way, that your travel plans were interrupted. That happened to a lot of people, unfortunately, this week. But one of the things that I think I may have missed is kind of the modes of transmission of this virus. And a question I would have is whether there's maternal-fetal transmission. In other words, are people born with it? Have people investigated that? And also about what the disease actually looks like in its acute stage. I know what a lot of adenovirus diseases look like, but is this like a classic adenovirus infection, or, you know, are people ill acutely when they have this infection? What do we know about it?
Good questions. Let me answer the second question first. So the adenovirus infections are generally classified into 2 or 3 categories. Some adenoviruses, the earlier ones, such as 82, they are linked with upper respiratory tract infections, and then there are some with conjunctivitis and some with GI disturbances. This particular virus, there is no specific information available about what exactly this virus does in people. There are many adenoviruses that we don't know a lot about what they do, although they have been identified. And the only clue that we have is that the first time it was isolated from a girl that was suffering from enteritis. So, and it may be one of those GI disturbance viruses. That's the clue. We don't know anything about it. In animals, it does not produce diarrhea. It does not produce any other major symptoms. In terms of maternal to child transmission, we would like to do that experiment. We have not been, we wrote a grant, didn't get funded for that one. So I don't know that. We have a question for that, question unanswered. In terms of children,
but may I ask, couldn't people study maternal newborn pairs?
Yes, yes. And
I don't think you'd have to do it experimentally.
I have not seen that paper, and we have not done it. We would like to, but very much like to. But we see in children at very early age this infection. Actually, children are the best model for us to study AD36 and obesity. It appears that there are many studies that have been done, about 7 or 8 studies by different research groups, I think, that have looked at infection in children, AD36, and its association with greater obesity, greater body weight. And it stands out very, very nicely. I think we all acquire a lot of adenoviral infections in childhood, so this may be when you get it, but perhaps it is more remarkable early in child, the difference between the infected and uninfected group in terms of body weight.
Okay.
Because if you grow up to be 45 or something, there may be other influences that are working on your body weight, and so that difference may not remain as clear. But in children, the effect is remarkable, the difference, rather.
And we have one other question, and then we're going to move the next speaker, Dr. Birnbaum.
Linda Birnbaum, Director, NIEHS. So this actually was pretty eye-opening to me. I appreciate the talk. I had very little information on the idea that infectious agents could play a role in the obesity epidemic. It's clear that obesity is an inflammatory condition, and I guess I'm wondering, so that's one of the things that would be interactive here. And the other thing is we do know that viral exposure can alter susceptibility for infectious agents. So do you think that there's a possibility, or it might be something we should design some studies to look at, co-exposure between infectious agents and environmental chemical exposure?
That's an excellent question. Yes. So, and on not only at chemical level, but it could be even, say, for instance, gut microbes. There are many adenoviruses, for instance, have been isolated from the gut, or those who are immune compromised people, for the first time they have been isolated. So viruses can play a role in both directions, either virus influencing, say, for instance, chemicals or gut flora, or gut influence, bacteria influencing viruses. It can go in either direction. And there are studies, I just don't have the time to explain all those, that indeed many viruses act in collaboration with various other factors, and only then they express their phenotype, which is what I was referring to as stars lining up. And there are studies that have been published about describing that, just exactly that effect.
All right. Well, thank you so much. And we will move to a panel discussion at 10:50. So hang on, and we'll include you in that online. But we're now going to move to Dr. Bailey. Your slides are up, and so I want to invite you to begin your presentation.